Hello, everybody. My name is Phoebe Jekielek. I'm the research director at the Hurricane Island Center for Science and Leadership in Penobscot Bay, Maine. Today, I'm going to talk a bit about the history of both wild and aquaculture scallop industries in Maine and how we're working to use eDNA tools as a way to assess both. In addition to being the director at Hurricane, I'm also a PhD student in the Maine eDNA program highlighted at the beginning of this session, and I'm working with Dr. Heather Leslie at the Darling Center and Dr. Nichole Price at Bigelow Labs. A little bit about Hurricane. We're located 12 miles off the coast of Rockland, Maine, in the Fox Island Archipelago in Penobscot Bay. This view gives you a good idea of our location within the archipelago itself, our distance from shore, and our location within Penn Bay. Hurricane is a 501c3 nonprofit with a mission to integrate science education, applied research, and leadership development through year-round educational programs in a seasonal, sustainable island community. Our research agenda focuses on answering local high-priority questions and increasing scientific capacity regionally through collaboration. We manage a 3.2-acre research aquaculture farm to the north of the island, where we currently grow scallops and kelp for research purposes. Since 2013, we've been working with local wild scallop harvesters, monitoring populations, and in 2016, we began growing our own scallops. So we've had our hands in both wild and aquaculture work for a number of years. I'm going to give a short overview of both of these industries. Maine has long supported a lucrative inshore dayboat scallop fishery. This fishery is managed in 3 zones along the coast of Maine, using size and catch limits, limited access areas, targeted closures, and most recently, rotational closures. Scallops are unique among their bivalve counterparts in Maine in that they have a strong wild population and commercial harvest, and now they also have a scallop aquaculture industry developing in parallel. So sprinkled throughout these management zones we just saw are now aquaculture farms. Just to be clear, not all of these dots on this map are scallop farms, but 193 of them could be someday. Currently, there are 193 total leases or LPAs listing sea scallops to grow, with another 7 in process. 49 of them are actively growing scallops. As these numbers show, there's a lot of growth potential for the scallop aquaculture industry, which is very exciting. And this potential and the co-location of farms within management zones sets us up for a unique opportunity to explore interactions between cultured and wild populations, the potential biological and ecological effects of growing scallops in aquaculture, and potential impacts of a changing climate on scallops as a whole. So a unique tool to evaluate these dynamics can be found in the use of environmental DNA, or eDNA. The Maine eDNA Sustainable Fisheries theme and the Larval Black Box Project is charged with evaluating larval dynamics of commercially important species. Specifically, we're to disentangle the larval supply side from the post-settlement side of shellfish recruitment ecology to improve sustainable use of wild and cultured resources. We do this by relating scallop and mussel gamete and larval densities to eDNA concentration as a function of local broodstock abundance, spawn timing, and oceanic conditions. This goal was deeply informed by previous and influential work led by Skylar Baer, Pete Countway, and Rick Wally, and has set the stage for success to address it. Some of the specific driving outcomes from this work include the development of scallop-specific primers and probes based on ITS gene fragments, which allow us to confidently identify scallop eDNA from other shellfish eDNA in water samples and plankton toes. Using these specific probes and primers, the team established a gene copy number determined through quantitative PCR or qPCR primers and size fractionation protocols can be used as a proxy for sea scallop gamete number, specifically sperm in the water column. There's near perfect agreement between sperm cell count and eDNA copy number using a laboratory calibration as shown in the figures above. So what are the next steps in utilizing this unique tool to address the goals of the Larval Black Box Project? First, we plan to conduct more rigorous field testing on farms and around wild populations to ground truth the use of eDNA tools to detect spawning events and larval production in cultured and wild populations. Goal 2 will conduct mesocosm experiments to determine exogenous eDNA generation rates from wild and farm populations. Goal 3 includes understanding whether or not targeted water sampling at various depth strata and during various points of the spawning season can be used to spatially isolate gene copy number from adults and gametes or larvae. By addressing these 3 goals, we can carefully construct sampling designs that could allow this eDNA tool to be used both for adult stock assessments, to indicate the presence, magnitude, and timing of scallop spawning events, and to estimate recruitment potential. So where are we at with these goals? Luckily, we've already been collecting data to address goal 1. We've been using the Hurricane Island Aquaculture Farm site at Site C and partner with 2 additional Scallop aquaculture farms in Penobscot Bay, one on North Haven Island, Site B, and the other on Andrew Island, south of Stonington in Site A. We monitor spawning and conduct eDNA sampling at each of these sites. To monitor spawning, we use gonadosomatic indices, or GSIs, which is the proportion of the gonad to the rest of the soft tissue. This method has been used to assess spawning in scallops in various studies and areas, including here in Maine, and represents an approximation of the potential spawning magnitude and is used as an indicator for spawn timing. Scallops are sexually dimorphic, so we're able to easily determine males from females. Weekly sampling occurs at each farm in June through mid-September. And at each sampling date, we dissect all scallops for meat weight, total viscera weight, gonad weight, shell weight, and shell height. In addition to collecting scallops for GSIs at each site, we conduct vertical plankton tows, approximately 1 meter off the bottom to the surface, to detect scallop larvae and eggs. A vertical profile at each site with a sonde housing and an optical dissolved oxygen, pH, fluorescence, and conductivity temperature probes. We also collect 3 liters of water to be filtered into 3 1-liter samples and later extracted and quantified using qPCR in order to assess for scallop larvae and gamete presence. The plankton tows, vertical profiles, and Niskin water collections are also conducted at a site distant from the farms to act as controls. To take just a quick look at some of the results we have to date, this figure shows a time series for the GSI sampling at the 3 different sites. A spawning event is indicated by a peak in the GSI, as you see here, and then a subsequent decline. The GSI sampling informs our understanding of scallop spawning and allows us to explore differences we might see between sites. It also ground truths our eDNA sampling. If we see a spawning event, we expect this to be reflected in our eDNA analysis and see a spike in the presence of gametes or larvae. So this is just a table showing the number of samples collected, filtered, extracted, and amplified from each farm and distant sites so far. All of this work happened in 2020. So far, we've collected a total of 177 samples, with 24 analyzed through to qPCR. Some filters may also be used in the future for metabarcoding analysis to assess food availability in farms. So as you can see, we've got a way to go, but we're on our way. To address goal 2, we will conduct mesocosm experiments using the Bigelow Lab seawater facility. Little is known about just how much DNA is being shed by scallops in general. So we'll set up mesocosms to quantify eDNA generation of different scallop abundances to simulate both wild bed and farm conditions. The suite is a continuously flowing seawater system with both filtered and raw seawater access and temperature control. This will provide us the ability to test shedding rates at different temperatures for each treatment as well. We expect eDNA signals to increase with increased abundance in wild and farm simulations, to be higher for scallops and gear, and to increase with increasing temperature. We are still in the process of planning this experiment, but we hope to conduct mesocosm experiments in fall 2021 or winter 2022. Lastly, for goal 3, we actually conducted a pilot experiment last week to address goal 3, the vertical and horizontal sampling above wild beds and aquaculture farms to detect differences in scallop eDNA signals inside and outside of spawning seasons in wild populations and on farms. At a known wild site off of Hurricane Island, we collected water samples throughout the water column at approximately 3-meter intervals, starting at 1 meter above the bottom. At the Hurricane Island Aquaculture Farm, we collected samples starting at 1 meter distant from a lantern net, and then at distances exponentially increasing out to 256 meters. This pilot experiment is part of a group project for a class and will inform future sampling and experimental design. We expect that the vertical and horizontal scallop eDNA signals will vary according to seasons, with temperature, and with tides. So the next steps, we have more filtration, extraction, and quantification of vertical and horizontal samples in addition to our eDNA sampling at aquaculture sites, which we will continue weekly through September 2021. We will continue the development of sampling design for goal 3 for the summer-fall 2021 data collection and mesocosm experiments. And we'll incorporate some of this data, including the GSI data, into predictive modeling of potential larval dispersal from farms using high-resolution hydrodynamic modeling. Thank you so much for your time. And I do want to take a moment to acknowledge our collaborators, our farmers, Marston and Bobby Brewer of Penn Bay Farm Scallops. You can find their products online at pennbayfarmscallops.com, as well as a cookbook that they've developed to educate about aquaculture scallops. And also Hannah Twombly and Connor O'Neill from Maine Island Aquaculture in North Haven. I also want to acknowledge Madison, Renee, Jen, Sarah, and Jason for all the work that they've contributed to these projects. And the funding sources, Davis Conservation Foundation and NSF EPSCoR. Please feel free to reach out to me at either of those emails, and I look forward to hearing from you. Thanks so much.